Alaikum. Welcome to Advances in Microbial Pathogenesis and Biotechnology. This is an advanced course that touches the pathogenesis, mechanism, and factors influencing infection and prevalence of those infections among the general population. So this is second part of the course, and it goes to focus more entirely on emerging and evolving infections that centered around bacterial, viral, fungal, and parasitic pathogens. We are going to give much emphasis to factors, mechanism, and public health impact of those emerging pathogens and how they are distorting the normal well-being and also the economic of the world, specifically Nigeria or West Africa in particular. Throughout history, from the Bronze Age to the Stone Age down to the Industrial Revolution, several civilizations have gone through a lot of weather outbreaks that killed millions of people. Some of these infections we are able to cure them or eradicate them, or they were wiped out by globalization or weather or a lot of other things. HIV have been serotypes, and there are a lot of biotypes that are re-emerging, and some they are just evolving. That's why we have several biotypes and serotypes of HIV. Of recent, we have cases of Ebola, which initially it manifested in some certain places and it sits down, but eventually it is re-emerging again. We also have cases of Zika, but the most prominent of all is the COVID virus, which initially was a SARS virus, if you recall, during the time of Obasanjo. A lot of chickens were slaughtered due to outbreak of SARS. Then after some certain years, the same class of virus manifested in the Middle East, which we call it the MERS virus. It have the same symptoms with the COVID-19. But the trends show that this COVID-19 metamorphoses and changes due to mutation from SARS to MERS virus and to COVID-19, which by the end of the pandemic, we also have other strains like COVID-20 and also 21. So these are evolving infections. We have such kind of cases in bacteria, we have Legionella disease and also Lyme disease. And which is also negligible is the fungal infection. You hardly find cases of infection caused by fungal. These are emerging infections and they are also evolving. There are peculiar situations and factors that they are speeding and driving this evolution of pathogens and incidences related to these pathogens. But to be clear, these kind of pathogens will continue to be evolving and changing due to mutation in their molecular makeup and also genetic gene transfer, as we are speeding it up via globalization, traveling, weather, and a lot of other factors. Now, re-emerging infections are infections that they have been there over 20 years or 30 or 50. In West Africa specifically, we have been suffering from typhoid, malaria, tuberculosis, cholera, measles, and dengue. You can list them a lot. They are there, and we are fighting them. They are driving the economy, and they are also endangering the economy and lives of individuals throughout the world. But most importantly, specifically in West Africa, due to our weather condition, congestion, urbanization, globalization, and a lot of other things. But the bottom line here is that this evolution and emergence of these infections is usually centered around our behavior. Now, as I've said, all this is centered around the factors that were affecting the emerging and emerging infection. If you look at how we are behaving with drugs and antibiotics, we are creating an avenue where people are misusing and overusing drugs. If you are taking drugs and you are minimizing the concentration of the drug at the site of infection, you are convincing microorganisms to develop resistance because they are dealing molecularly molecularly with enzymes they produce in degradation, and also mechanism for resistance. As well, when you are overusing drugs, you are creating an avenue where other drugs cannot dwell. So this is also contributing in the case of re-emergence of a lot of other infections. As well, globalization and rapid traveling. Now, our world is a global village where whenever and however you want, you can travel across a lot of continents and countries, and you can take an infection knowingly or unknowingly from one part of the world to another part of the world. And wherever you go, whenever you change from one location to another, you are changing from one weather condition to another. That's how globalization and also climate change, invariably in the case of emergence and evolving infection, they are going hand in hand. Climate changes is something that we are experiencing usually after a decade, major change, and after some certain years, minor changes. You see that the weather is changing. And this is creating an avenue where this microorganism they can redwell again, because as a certain change of weather is creating a conducive atmosphere for this microorganism to reemerge again. Overcrowding due to development in the area, civilization and industrialization, people are leaving their villages and they are teeming out and gathering in a city center. So the metropolitan area, whenever there is gathering, there is congestion, there is a lot of aerosols, there are a lot of gutters, there are a lot of that everywhere, and this is creating a more good situation for microorganisms to dwell more, and eventually some of them will evolve due to mutation. As you know, when there are a consortium of microorganisms in a particular place, they evolve together due to horizontal gene transfer. So this is something that is contributing in the emergence and evolving of these infections. But much important factor is the fact that the rise in the immunocompromise or immunosuppressions of the general population. A lot of people, due to their etiquette and lifestyle, their immunity is suppressing gradually, is decreasing in strength. People are smoking, people are drinking, occupational hazard, and a lot of other things is reducing the strength of the immunity. So a lot of people are no longer immunocompetent, they are immunocompromised. And this is creating a good situation for these emerging and evolving pathogens to enter into the human body and manifest and colonize a lot of tissues, either in a localized infection or more secondary infection where a lot of tissues and organs would be affected. Now, antimicrobial resistance, as we know, it has been categorized by so many researchers. But the bottom line here is that we have three, which are defined by how many drugs a particular pathogen is resistant to. In the case of MDR, which means multi-drug resistant, is a kind of multi-drug resistant where 2 or 3 categories of antibiotic within one class of antibiotic the pathogen is resistant to. 3 types of drugs within the beta-lactamases, it is an MDR resistant pathogen. Whereas if a particular pathogen is resistant to 2 or more classes of antibiotic, it is an extended drug resistant pathogen. But in a situation where a pathogen is entirely resistant to all classes of antibiotics, it is termed the pan-drug resistant pathogen. So this is a resistance that we are recording virtually every year in and out. And now most of the pathogens, be it either evolving or emerging, they are pan-drug resistant pathogens because they are resistant to drug resistant genes and also virulence genes. That's why they are very dangerous. And they are acquiring this resistance through one horizontal gene transfer, either via conjugation or transformation. Most recently, as you know, during the time of the pandemic that caused by the COVID-19, there are a lot of cases where people become affected and more acutely infected by the COVID-19 because they are already resistant to a lot of drugs and antibiotics. That's why you can't entirely exonerate antimicrobial resistance whenever you're talking about evolution of infection and also re-emergence of a lot of other pathogens. Now, in the case of climate change and also ecological impact on the emergence of infection vectors, parasites, they are feeling much comfortable to dwell and transfer pathogens from one host to another. So there are a lot of vectors that were migrated because of change in weather. As well, now people are taking pets as luxurious animals, and eventually the transfer of pathogens can go from animal to human being. So there is zoonotic spillover. People are kissing pets, animals of all sorts, and this can transfer pathogens from an animal to a human being. And you know, microorganisms are so strange to the extent that they have the capability to adapt to virtually all kind of weather. So even in the environment, a lot of weather evolving pathogens, they feel convenient over the change of the climate, and eventually they can adapt to the situation, which will help them in effectively infecting a lot of horses from human being to animals. They have the ability to change this by changing temperature from yeast to mold. That's why a lot of fungi like the Candida auris, they have this virulence adaptation. One of these new and emerging bacterial infections include multi-drug-resistant Acetobacter baumannii, and we also have Mycobacterium tuberculosis. A lot of other hypervirulent strains. If you take an example of hypervirulent Klebsiella pneumoniae, which is affecting a lot of people to the extent that where you cannot differentiate the infection by hypervirulent Klebsiella pneumoniae and hypervirulent Streptococcus pneumoniae. So that's why we have a lot of cases of infant pneumonia and also adult pneumonia. But these pathogens, they are acquiring their resistance, as we have explained, via recombination and mutation, and they are also acquiring virulence factors whenever they are growing in a consortia of microorganisms. When these bacteria acquire these genes, they can either acquire a drug-resistant gene, which you know, like the beta-lactamase drug-resistant genes, and a lot of other drug-resistant genes, but as well they can acquire virulence genes. Now, when they are living in either biomass or in biofilm situation, they have to produce adhesion factors like adhesins, which they can also acquire via genetic gene transfer. And this is also helping them in increasing their virulence and evading drugs in the human body. There are a lot of other situations where these pathogens, they can evade infection. One of them is that antigenic variation. We know that they have a lot of epitope on the surface of their body, but for them to evade immunity, they create other types of antigenic binding site. So the immunity can no longer recognize them because they have altered the binding site on their surfaces. Number two is that they have what is called molecular mimicry. Molecular mimicry is a situation where they can create a protein which is similar to the body. So the immune system can no longer recognize them because they have mimicked a particular protein which is self. So the immune system cannot recognize what is self. Now, after all, whenever there is emergence or re-emergence as a result of drug resistance or mutation or immune evasion or a lot of other things, the public will suffer from these consequences. At the end, when you are infected with such kind of pathogens, you find that the patient has a prolonged morbidity where the incubation period is very short, but the infectivity is very long. People are focusing more in antimicrobial stewardship to make sure that we have created alternative antibiotics and drugs that can fight these evolving pathogens. Now we are envisaging to create a synergy between synthetic drug, organic drug, and nanoparticles, and also we use a lot of other organic substances to extract some certain component or bioactive component that they have the efficacy to prevent such kind of evolving and emerging infection. Antigen-antibody reaction, in the case of, let's say, now malaria can be rapidly diagnosed using strip tests, as well typhoid, as well H. pylori, and a lot of other antigen-antibody reaction. They are extremely effective in making sure that we have detect the causative agent. Then we have to know the prevalences and the rate of incidences in a particular test population. Then gradually we can check and see if some certain available cheap bioactive component can be embedded in synthetic antibiotic to make sure that we create a synergy in a particular drug that can effectively deal with this evolution and emergence of pathogen. Now, most of the drugs that we are using is they are just nucleotide reverse transcripted inhibitors, because if you inhibit the rate of reverse transcriptase during the replication cycle of the HIV patient, HIV virus, the virus can no longer replicate it, because you have inhibited the most important line of translation and transcription. The fact that the HIV virus is a reverse transcriptase enzyme, the only way that you can stop it working is by inhibiting the rate of its replication. That's why the NRITS and these drugs, they have the capability to inhibit the process. So if you recall the central dogma of molecular biology, where there is a presence of DNA, then transcription, translation, then the protein is decrypted by the ribosomes. In the case of viruses, there is a DNA, there is the RNA, because it is an RNA virus. So since it is an RNA virus, it requires reverse transcriptase to a DNA. Then from the DNA, it will transcribe again, then translate it, then produce a protein. So for you to stop it, you have to stop the reverse transcriptase. So all these drugs, the protease inhibitors, the integrase, and the entry inhibitors, they all serve in a situation that they can stop that process in such a way that the HIV virus cannot continue multiplying. So of reason, we have a lot of drugs, but some of them, they are no longer working. So the lenacapavir, which is capsid inhibitor, it's just a phenotype inhibitor, unlike the other reverse transcriptase inhibitors. Initially, they are long-acting drugs, but gradually they are suppressing, and they are meant or produced to make sure that they are effective against those drug-resistant HIV viruses, but due to mutation, they are also receding. So not only bacteria, virtually all these are evolving and emerging pathogens. They are developing resistance. If it is a fungi, they are developing resistant to fluconazole and ketoconazole. If it is a bacteria, it is developing resistant to technically all the antibiotics, as well the viruses, they are also developing resistant. Now, there are also factors that are affecting fungal emergence, but they are technically the same. But most importantly, as I've said, is immunosuppression and climate change. So they are all the same across all pathogens, because this is what is affecting not just our pathogens and diseases, but also the economy of the world. So they are technically the same, but it goes across different pathogens.